I have to confess to stupid professor syndrome today. I was uploading the highlights for the last lecture in the schedule page, and I spent some time, and I put it all together, and I was very happy. And I clicked send because you have to upload. And what I didn't realize was when it's sent, I hadn't had my individual file of the thing selected. So not only did my individual file go, but the entire directory of files went up and replaced the files that I had online. So all the highlights have gotten scrambled. So if you go back and look at the highlights right now, which I don't recommend doing, you're going to see highlights in some cases from this year, in some cases from previous years, in some cases just scrambled. Central Web Services is working on it, and they hope to be able to recover it. So I'd recommend the only highlight that's up there right now that you can count on is the one for last lecture. Okay, so that one is actually real and good. I wouldn't recommend downloading other highlights until I send you an email saying, okay, it's been fixed. And if they don't fix it, then yours truly is going to have to do a lot of work that he hopes he doesn't have to do. So we'll see how that goes. So that'll teach me to not be paying more attention to what I'm doing. What's that?
I just replaced all of mine.
You just replaced all yours this morning? What time?
Just now.
Just now? Well, then I wouldn't go back and use those highlights because you're going to see things from previous years. And though many of the things are the same, they're out of sequence. And some things I don't talk about, like Maldi-Toff, you'll see from previous years and stuff. And I haven't talked about it this year. So sorry about that. All right. So my Friday didn't get off to a real good start, I guess I'm saying. So I hope yours is going better. Let's see. We have an exam in here a week from Monday. And if you haven't downloaded the practice exam I have online, you should do that just to get an idea about what my format is. The format will be exactly the same as what's on that exam. Please, please, please don't use old exams as a study guide. Not a good idea. I don't put them up there for that purpose. I put them up there so that you see the format of my exams. Things change from one year to the next. You waste time on things that don't make sense. And using it as a study guide is just not a good idea. It's better to study the material, study the problems that I've given you, study the highlights and things like that. Using old exams as a study guide is a sure prescription for disaster. So please don't do that, okay? But anyway, do look at the old exam for the format, because the format will be the same. On my exams, you'll see 3 sections. First section will be relatively short answer. Second section will be problem solving. And the third section will be longer answer. And so that's the kinds of things that you can expect to see in there. Okay, and I'll say more about exams starting next week. Now what I want to do here is finish up talking about hemoglobin. Last time I got started talking about fetal hemoglobin. And fetal hemoglobin is interesting, particularly compared to the maternal hemoglobin. And so let me just say a little bit more about that and then also sort of answer the question that I posed to you about fetal hemoglobin. And I really don't like that battery doing that. Let's see if I can maybe change the battery and see if that makes a difference. Doom. We'll see if that makes a difference. Sometimes it does. All right. So fetal hemoglobin, the fetus has a challenge. So the fetus has to get oxygen, and to get oxygen, it has to take it away from mom. And mom's hemoglobin kind of lacks oxygen as well. So I said that there was a difference in fetal hemoglobin from mom's hemoglobin. Maternal hemoglobin, as any adult hemoglobin, has 2 alpha subunits and 2 beta subunits. In fetal hemoglobin, the 2 beta subunits are replaced by 2 related subunits called gamma subunits. So in fetal hemoglobin, you've got alpha 2 gamma 2 instead of alpha 2 beta 2. And the difference with that is that in alpha 2 gamma 2, what you see is that there's not a place to bind 2,3-BPG. Okay? There's not a place to bind 2,3-BPG. So that means then that the fetal hemoglobin really can't flip into the T state, at least not very easily, not very well. That means that the fetal hemoglobin ends up being in the R state, and the R state is the state that likes to bind oxygen. Now, That turns out to be useful to answer the question I posed for you at the end of the last period. It turns out to be useful because that means that the fetal hemoglobin is going to have a higher affinity for oxygen than mom's hemoglobin is going to have, and it is in fact going to take oxygen away from mom's hemoglobin. And that's good. But because it doesn't do as much flipping, for example, when there's 2,3-BPG around, then this fetal hemoglobin can't bind 2,3-BPG and let go of oxygen. It means that fetal hemoglobin has a harder time letting go of oxygen. Now, my sort of question at the end of the last period was, do you envision this being a problem? Anybody? Problem? Yeah, Kyle?
Clarification first. Do we have 2,3-BPG crossing the placental wall? And you said it's not in the...
Yeah, so the question is, can 2,3-BPG cross the placental wall? I would guess that it can.
It doesn't do anything.
Well, it's not going to affect fetal hemoglobin, that's for sure. Okay, let me ask you another question that students frequently ask me. If mom's a smoker, okay, and she's loaded with 2,3-BPG, and 2,3-BPG does cross the placental wall, then what's happening to that fetus? What's that? Not from 2,3-BPG, because the fetal hemoglobin doesn't do anything with the 2,3-BPG. All right? The fetus does get less oxygen, but only because mom's carrying capacity for oxygen is lower. All right? So if mom's carrying capacity for oxygen were normal, which it's not, okay, then the fetus would have, the smoking would have no effect on the fetus, even if the fetus were loaded with 2,3-BPG, because the fetal hemoglobin doesn't bind that. Now, to get back to my question, do you imagine that 2,3-BPG would pose this high affinity for hemoglobin poses a problem for the fetus? The answer is no. And the reason it's not a problem is the fetus isn't out exercising. Except for kicking mom, the fetus doesn't have large differences in demand for oxygen. That's why we need it. When we have actively respiring tissue, our actively respiring tissue is producing 2,3-BPG, binding the hemoglobin, causing oxygen to be released. The fetus isn't out jogging. The fetus isn't out lifting weights. The fetus isn't out stressing about its day. All right? All these things cause effects physiologically inside of adult people. So the fetus is not affected in that sense by that 2,3-BPG. Okay, let's see. So that's the story about fetal hemoglobin. Now what I want to do now is turn our attention to talking about yet another phenomenon associated with hemoglobin called the Bohr effect. And the Bohr effect is a generalized effect, and it's also related to the release of oxygen by actively respiring tissues. And it is caused by yet something else. So let's take a look at the Bohr effect again from our graph that we've been showing before. So on the y-axis, we see, again, hemoglobin now being plotted. We see what fraction, what percentage is saturated with oxygen as a function of oxygen concentration. We see two different curves here. We see the binding curve for hemoglobin at pH 7.4, which is the top curve, and we see the binding profile for hemoglobin at pH 7.2, which is the bottom curve. You'll notice that at pH 7.2, the affinity of hemoglobin for oxygen is lower. Now, this makes very, very, very good sense from the perspective of physiological need and respiration. Just like when we have actively respiring tissue, we have 2,3-BPG being produced, so too when we have actively respiring tissue, do we have protons being produced. Okay? Protons being produced lower the pH in that local region of the place where the tissue is actively respiring, and again, cause hemoglobin to have a lowered affinity for oxygen and release oxygen. So both in the sense of 2,3-BPG and in the sense of protons, which are both indicators of active respiration, the hemoglobin is primed to give up oxygen to the tissues that need it. Okay, so that's pretty cool. Now, the question is, how does that happen? All right? And it turns out that the way that happens is the byproduct of protonation of hemoglobin. The protons that are in higher concentration in the actively respiring tissues are actually taken up by histidine residues inside of hemoglobin. They're taken up by histidine residues inside of hemoglobin. All right? This shows a histidine residue. This shows a proton being taken up as shown here. And the result of that is something that looks like this. Now, I'm sorry, I'm jumping a step ahead. If we look at the guy on the left, this is a schematic representation of that histidine that's gotten that extra proton. So the histidine has the extra proton. So it's in a low pH. It's grabbed the proton. It's got the proton. And it turns out that when carbon dioxide comes along, actually, I really am getting ahead. Okay, let me back up. I'm getting ahead to carbon dioxide, and I don't want to do that. Okay. The second phenomenon related to the Bohr effect, and all these things that I'm talking about here are the Bohr effect. So first of all, I've told you that oxygen is released. And it's released as a result of that change in pH, and that pH change happens when histidine takes up a proton. You can imagine that histidine taking up that proton causes a slight conformational change, which is again favoring the release, not the uptake of oxygen. And that's exactly what happens. Another aspect of the Bohr effect is the fact that hemoglobin will actually take up carbon dioxide. Hemoglobin will bind to carbon dioxide. Now, I want to underline something here. Hemoglobin does not, underline not, bind carbon dioxide at the heme group. It binds it at another place in the molecule. And that other place in the molecule is the histidine that got that proton. Okay? I didn't point out before, but I should point out here that there is something else besides oxygen that will bind to the heme group. And it's very important and it's very nasty. And it's carbon monoxide. Carbon monoxide will compete with hemoglobin for binding to the heme group. Carbon dioxide will bind to hemoglobin, but not at the heme group. And in order for carbon dioxide to bind, it has to find a protonated histidine. Okay. Well, this figure simply depicts the fact that actively respiring tissues, in addition to producing protons and in addition to producing 2,3-BPG, also produce carbon dioxide. Okay? Carbon dioxide, as I'm sure you all know, is a byproduct of respiration. The more respiration, the more carbon dioxide. The formation of carbon dioxide and the release of carbon dioxide is actually one of the sources of the protons. Carbon dioxide released into an aqueous solution in the presence of an enzyme, we'll talk a lot about this term, called carbonic anhydrase, makes carbonic acid, which releases protons. Okay? So that's one of the reasons that actively respiring tissues are producing protons. If I look at this phenomenon of the Bohr effect now in view of both protons being produced and carbon dioxide being produced, what do I see? Well, the top line shows hemoglobin binding at pH 7.4, as you've seen before. The next line down, which is the blue line, shows you the same hemoglobin in the presence of no carbon dioxide, just as the top one was, but at pH 7.2. So that's what you saw earlier. Now if you add carbon dioxide and that low pH, look how the curve shifts even lower. And again, actively respiring tissue is producing protons, it's producing carbon dioxide, and it's lowering the affinity of hemoglobin for oxygen. Okay? So all of these things are contributing now to the release of oxygen where it's needed. Yes, sir?
Could it carry for oxygen and the CO2 then?
Could the hemoglobin carry 4 oxygens and the CO2? In theory, it could. But as you start putting on protons, you're already favoring the release of oxygen. You've already got 2,3-BPG there. So the reality is it probably wouldn't be you wouldn't find very many that would have much oxygen and CO2. So it's a good question, but the reality is that would be difficult to have very much of that. Okay, now, getting back to where I was jumping ahead, the way that carbon dioxide is carried in hemoglobin is as follows. Here's the histidine with that extra proton. Carbon dioxide comes along. It will displace that proton, and form a carbamate a complex shown like this. This is actually a covalent complex, and this gets carried back to the lungs. So now we've got actively respiring tissues. They're releasing carbon dioxide. Some of that carbon dioxide, I want to emphasize some, not all of it happens this way. Some of that carbon dioxide is forming a carbamate ion in a complex with histidine in the hemoglobin. And as a consequence, this is now going to be carried back to the lungs. The last part of the Bohr effect happens in the lungs. And the lungs, we basically reverse all of this process. And this figure is not a particularly good one. When you get to the lungs, you at this point have hemoglobin that has got carbon dioxide attached to it. It doesn't have hardly any oxygen attached to it. The pH of the lungs is not going to be as low as the pH was in the actively respiring tissue, so the pH will be higher. Okay? This will change the shape of hemoglobin slightly. More importantly, and perhaps most importantly, in the lungs, the oxygen concentration is very high compared to what it has been. The last part of the Bohr effect happens when oxygen concentration is high. It literally forces itself onto the hemoglobin. That changes the shape of hemoglobin. It helps to break those bonds with the carbon dioxide, so the carbon dioxide is released, and you exhale carbon dioxide as a consequence. Pretty cool stuff. So we've seen the production of carbon dioxide, the carrying of carbon dioxide to the lungs, the forcing off of carbon dioxide from hemoglobin in the lungs, and the exhalation of carbon dioxide as a consequence. Is that a question, or is that? No, not a question. Okay. Okay, does that make sense? Questions? Yes?
So did you say that the carbon dioxide is released because of the change in shape when the oxygen is bound to the hemoglobin?
That's correct. So oxygen literally forces its way on. Yep. Okay? The hemoglobin changes shape as a result of that, and all these combine to help favor the release of carbon dioxide.
What about that proton?
That proton has to be picked up from the solution. And that's another reason you see the pH going up. So the protons are being taken from the solution. Yes?
Is the 2,3 BPG there at the same time that CO2 is?
Is 2,3 BPG present at the same time the CO2 is present? There's nothing to stop the two from being present at the same time. So the binding of 2,3 BPG doesn't have any effect on the carbon dioxide, only the heme group where the oxygen is. Okay? Good question, Marcia. Yes? So her question is the CO2 produced by the tissues part of what causes the red blood cells to produce protons? Is that what you said? Yes, because that forms carbonic acid. Yes. So those protons that are released by the carbonic acid are what lower the pH and favor the release of oxygen at that point. That's correct. Make sense? Okay. Yes? Okay. So the proton, I'm not sure I understand the question, but proton, we're in a solution that's got a buffer. So proton concentration is maintained relatively constantly. There are always protons available if you need to replace something with a proton, or you just need to simply give a proton. So I'm not sure I'm answering your question.
Well, the proton that was picked up by the histidine in the hemoglobin, is that then like released once the oxygen
Okay.
binds?
So when hemoglobin released a proton on the binding of the CO2, that proton can be just buffered by the buffering system of the body. Okay? Okay? Yes, Lori.
The hydrogens that are released through the CO2 turning into acid and then
Uh-huh.
releasing the oxygen, are
Right.
those the hydrogens that are then bound by the histidine to bind the CO2 and take it back to the lung?
So that's a source of protons for that, okay? We always want to think of a one-to-one relationship. This proton goes over here, but we have to keep in mind that there are billions of protons floating around the bloodstream that came from any variety of sources, right? So there's protons available to have that happen. Okay. Well, as if there wasn't enough with hemoglobin, I have one last consideration for hemoglobin, and it's kind of cool too. It's not cool if you have this disease, but it's cool knowing what you know about hemoglobin to actually be able to treat this disease. Sickle cell anemia is a disease of hemoglobin. And it's a disease that there are many different forms of the disease, some much more severe than others. Many of the forms affect the beta subunit of hemoglobin. And when a person has sickle cell disease is a genetic disease. You can't catch it as such. You either were born with it or not. What happens is the hemoglobin in the blood cells of sickle cell anemia have, in some cases, as simple as a single change of a single amino acid. And that single change of a single amino acid doesn't affect hemoglobin's ability to bind oxygen, contrary to what you might think. Okay? What it does is it affects the structure of hemoglobin under low oxygen concentrations. So in sickle cell anemia, what happens is when you have hemoglobin traveling through the bloodstream and you get to a place where there's very low oxygen concentration, hemoglobin gives up its oxygen as it's supposed to do. But in the absence of oxygen, that hemoglobin now actually forms polymers with other hemoglobins. And these polymers, which don't form with normal hemoglobin, cause the blood cell to assume a shape that looks like a sickle. Okay, so blood cells, we always draw these nice little round ovoid things, and they're smooth on the sides, and that's literally what blood cells look like. The sickle is not round and ovoid. It's sticky. It has points to the end. And that causes a problem because when the sickle cell forms, it's forming in places where there are low oxygen concentrations. And where is that? In the capillaries. Okay? In the capillaries. Those sickled red blood cells get stuck there. When blood cells get stuck in capillaries, no blood flows through them. Consequently, the capillaries, which are embedded in the muscles that are generating the need for the oxygen in the first place, consequently, these muscles are starving for oxygen. So a person with sickle cell anemia will find that they can develop intense pain, for example, with moderate exercise. And the intense pain happens due to the fact that the muscle cells are literally starving to death for oxygen. It's called anemia because people who have this have low blood counts. That is, low counts of blood cells. Why does that happen? The body has a mechanism to destroy what it sees as damaged blood cells. Cells that are sickled look to the body like they are, in fact, damaged, and the body takes them out of circulation, even though there's nothing really wrong with them other than that polymerization. As a consequence, it's an anemia, which refers to, again, lower concentrations of blood cells. So sickle cell anemia is very debilitating. It can be an enormous problem. It can be fatal in some cases. And one of the questions is, are there mechanisms for treating it? And there are many different strategies for treating sickle cell anemia, one of which involves altering the person's DNA, which is not a trivial thing to do by any stretch. And we're a long ways away from doing serious genetic manipulation on humans, although there are some cases where we have done that. An alternative approach to that is to actually induce in these individuals the production of fetal hemoglobin. Remember that fetal hemoglobin is made as you're a fetus, and by the time you're about 1 year old, almost all the fetal hemoglobin that your body is making ceases production. The genes are still there. They're just turned off. And so one strategy is to take and design drugs that will actually activate the production of fetal hemoglobin, because the fetal hemoglobin can replace the bad beta subunits and, in fact, rescue the person. In some cases, this actually has been a strategy that has worked to a limited extent. So it's an interesting approach. How long-term or how big of a success it'll have is totally unclear. Yes? Yes. So they produce the 3 different types. In fact, I simplify the description a bit. Your body is producing at any given time about 4 or 5 different types of hemoglobin, but by far the alphas and the betas are the most predominant. But the question which I think is next is, well, do these get in the way of the betas or anything like that? And you could imagine that would be some complications. So it's still early in the process of understanding how this might work. But in some cases, it actually has given relief. Yes, Lori?
How would that cause the body to stop producing the bad ones?
It doesn't. The bad ones are still there. But now you've got a better chance by the fact that you've got more good ones. Yes?
So then would they still have issues with exercising? Would they have a field hemoglobin?
Yeah.
They produce the 2,3
Yeah. Again, the prediction would be you probably would have a reduced problem with exercise simply because you're going to have a reduced ability to polymerize and a reduced ability to make those sickle cells. But yeah, you probably still do have some problem with that. Yeah. Mark? Okay. Okay. All right. Well, I think we haven't had a song for a while, so it's appropriate at this point that we should have a song. And this one is designed to help you learn all about hemoglobin. It's to the tune of Santa Claus is Coming to Town, and it's called Hemoglobin's Moving Around. Please join with me or we'll have a pop quiz. Oh, isn't it great what proteins can do, especially ones that bind to O2. Hemoglobin's moving around. Inside of the lungs, it picks up the bait and changes itself from T to R state. Hemoglobin's moving around. The protoporphyrin system, its iron makes such a scene, arising when an O2 binds, pulling up on histidine. The binding occurs cooperatively, thanks to changes quaternary. Hemoglobin's moving around. It exits the lungs, engorged with O2, in search of a working body tissue. Hemoglobin's moving around. The proton concentration and high has a role. Between the alpha betas, it finds imidazole. That's histidine, by the way. To empty their loads, the globins decree. We need to bind 2,3-BPG. Hemoglobin's moving around. The stage is the set for grabbing a few cellular dumps of CO2. Hemoglobin's moving around. And then inside the lungs it discovers oxygen and ups the CO2 off to start all over again. So see how this works? You better expect to have to describe the Bohr effect. Hemoglobin's moving around. Thank you. Okay. It'd be nice to be able to stop there, but we can't stop there, so we have to go on. Oh, one more. Maybe if you guys are really good, I'll tell you a joke at the end of the period, okay? But I've got to have your full attention, you know, and I've got to have your interest and so forth. Yes, sir.
So my question, when that single change of a single amino acid, that conformation causes a polymer of those messed up amino acids.
So the question is basically what's involved in this change of the single amino acid in hemoglobin. This change of the single amino acid in hemoglobin, okay, changes the ability of individual subunits to interact with each other. I'm sorry, not individual subunits, but individual complexes of 4 to interact with each other. So they start associating with each other, one after the other, after the other, after the other, and that's the polymer I'm talking about, okay? So these are just associations, but they're long linear chains, and it's these chains that are causing that sickle shape to form. Does that make sense?
Yeah.
Okay. All right. Well, we turn our attention from something that's not an enzyme to something that is an enzyme. And we're going to see some parallels between what enzymes do and the way that hemoglobin behaved cooperatively. Okay, we're going to see some parallels there. Enzymes, of course, you know from any general biology class, are catalysts. Catalysts are things that speed up reactions without themselves being used up in the reaction. That's something you just basically learn. I mean, one of the earlier things I ever learned about biochemistry was that right there. An enzyme is something that speeds up a reaction and doesn't actually get used up in the reaction. We're going to see that enzymes are different than regular catalysts. That is, a chemical catalyst does not change. We're going to see enzymes actually change. during the process of catalysis. It's a transient change, but nonetheless, they do change. So we need to understand that. Okay? The enzyme carbonic anhydrase I mentioned earlier, it catalyzes the reaction that you see right here. Carbon dioxide plus water, and there's carbonic acid on the right. Carbonic anhydrase does something absolutely amazing. Okay? Absolutely amazing. And the amazing thing that it does is it takes this reaction and it will do this reaction at an incredibly high rate. Okay? Incredibly high rate. I'm going to give you some numbers on that in a little bit. All right? But it's phenomenal how fast carbonic anhydrase can make this reaction go. Okay? Enzymes, as I said, are different from chemical catalysts. And they're different from chemical catalysts not only in their structure, but also in their ability to speed up reactions by enormous amounts. Okay? This little tiny table, which is a little hard to read, but you can see up here, lists a variety of enzymes. And these enzymes catalyze different reactions. The reactions in the absence of an enzyme and how fast they proceed are shown here in this column. And the rate with which they occur is over here. So this is actually a rate uncatalyzed, this is a rate catalyzed, and this is a half-life, which is also another way of measuring how fast a reaction proceeds. The reaction catalyzed by OMP decarboxylase we'll actually talk about next term. If we look at that reaction in the absence of an enzyme, and we look at how long it takes half of the reaction to proceed to completion, it takes 78 million years for half of it to get there, okay, without the enzyme. That's a reaction that doesn't go very fast. Okay? If we look at the same reaction in the presence of this enzyme, we see that it catalyzes at the rate of 39 molecules per second per enzyme. All right? The actual rate is shown over here, and you compare the rate uncatalyzed, 2.8 times 10 to the minus 16th, with the rate catalyzed, 1.4 times 10 to the 17th. You see a difference there of 10 to the 33rd. This is an enormous, incredible enhancement of the rate of reaction of an enzyme. A chemical catalyst might be good to enhance a reaction speed by 10 to 100 to 1,000-fold. Enzymes routinely do it over 1,000,000-fold. Okay? So there's something very special about enzymes and the way and the rate with which they catalyze reactions. Here's our friend carbonic anhydrase. Okay? Well, whoa, it's way down here. Okay? But look at this catalyzed rate. This catalyzed rate is called kcat. I'll explain more about kcat in a little bit. Okay? Kcat is a measure of the number of molecules of product per molecule of enzyme per second. So for the OMP decarboxylase, that was 39 per second. For staphylococcal nuclease, it was 95 per second. For charismate mutase, it's 50 per second. Now, for carbonic anhydrase, this is a mind-boggling 1 million molecules of product per molecule of enzyme per second. That's pretty hard to fathom. I'll repeat that. That's 1 million molecules of product for every molecule of enzyme every second. Can you imagine doing anything 1 million times a second? No, you can't. Okay? Well, maybe you can. I don't know. From that little titter going across the crowd, I wonder what you're thinking of. I don't know. But 1 million times a second, that's really serious computational power. Okay? It's hard to imagine that there's enough of the substrate, that is, the substrate is the molecule that the enzyme acts on. It's hard to imagine that there's enough substrate that can get to the enzyme that fast and get converted. But it does. Okay? So this is a very incredible thing. Enzymes are very incredible things. And you see they have different rates of kcat. 39 per second is pretty darn good. Okay? I think you'd have a hard time thinking of 39 things you could do per second as well. All right? So these are pretty phenomenal rates that enzymes are able to achieve. Enzymes, we're going to see, don't work on everything. They work on specific molecules. They work on specific structures because enzymes themselves have specific structure. We've talked about the importance of structure and function. If you change the structure, you lose the function. Enzymes catalyze reactions by binding to substrates. And substrates are simply the molecules that the enzyme catalyzes the reaction upon. They act on substrates, and they catalyze a reaction on them at a special place in the enzyme called the active site. The active site is where the reaction of the enzyme is catalyzed. The active site is usually internal, although not always, to the enzyme. It has a very, very, very specific structure. And it's for this reason that when we change the structure of the enzyme, we lose function. What's happening is when you change the configuration of that active site, there's a very good chance you're going to lose the function of that enzyme. Some enzymes are more picky about it than others. We'll see some enzymes that will work on multiple related structures, others that will only work on one. Structure and function is absolutely essential. This shows the proteolytic cleaving specificity of two different proteases. Proteases are enzymes that cleave peptide bonds, as we've seen before. Okay. One enzyme, which we've talked about already, is trypsin. Trypsin will cleave right here, okay, adjacent to lysine or arginine, and there's the bond it cleaves. There are other proteases that will cleave only if that's an arginine. These guys are much more picky about their active site than this one is. Okay? And we'll talk about how that works later. Okay. Enzymes don't do everything by themselves. Okay? You saw in the case of hemoglobin, which I said was not an enzyme, that hemoglobin itself needed the help of something else, and something else it needed was a heme group. Enzymes use other factors called coenzymes to help them to catalyze, to bind molecules, to do a variety of things. So coenzymes, if you want a definition, are non-amino acid components of an enzyme that help the enzyme to do what it does. Okay? Now, this is a table. We're not going to memorize the table or any of that sort of stuff. But you should realize, of course, there are metals that help in this process. We're going to talk later in the term about some of these factors up here and some of the effects that they have on enzymes. Biotin is one that helps enzymes to put carbon dioxide onto something. Whenever we have an enzyme that puts a carbon dioxide onto something, it usually has a biotin associated with it. NAD, nicotinamide adenine dinucleotide, is associated with enzymes involved in reduction and oxidation. And we'll see how that plays into things later. Okay. Well, before we talk about enzymes, I need to say a little bit about free energy. Gibbs free energy was probably something you had at the end of your freshman year of chemistry. And it was probably something you went, about. Now, we're going to talk more about Gibbs free energy later in the term. And we're going to talk about it in a little bit more depth later in the term. But before I do that, I need to just sort of bring you up to speed a little bit about Gibbs free energy. Gibbs free energy is a very useful thing because it gives us an ability to determine the direction of a reaction. So if we look at a reaction and we look at the change in the Gibbs free energy that occurs in that reaction, the sign of that tells us whether or not the reaction goes forwards, backwards, or is at equilibrium. Okay? Now, the Gibbs free energy can be calculated, and the change in Gibbs free energy can be calculated. The change in Gibbs free energy is shown by this equation right down here. Delta G, which is the change in Gibbs free energy, is equal to the standard Gibbs free energy, which is known as the delta G 0 prime. That's what that little prime designation is there. Plus R, which is the gas constant, times T, which is the temperature in Kelvin, times the natural log of the concentration of the products divided by the reactants. That is the equation. And you might say, oh boy, another equation. Well, remember I told you you're not going to memorize equations, so I'll give you the equations on the exam. Another thing I'll tell you is that this looks like it's very different, but in fact, it's not that different from the Henderson-Hasselbalch equation, which by now you all have mastered. Okay? The Henderson-Hasselbalch equation, I'll remind you, said pH equals pKa plus the log of salt over acid. Right? I said pH can vary. I said pKa was a constant for any given acid, weak acid system. And the log term was salt and acid. As salt and acid changed, so did the pH change, right? If we look at this equation, The change in Gibbs free energy is a variable. It changes from one situation to another. The standard Gibbs free energy is a constant for a given reaction, just like pKa was a constant for a given weak acid. Instead of a log term, we have a natural log term, but the rules are still the same. If products over reactants are greater than 1, that term is positive. If products over reactants is less than 1, that log term is negative. Okay? Now, so the same basic considerations we had for Henderson-Hasselbalch hold for the calculation of the Gibbs free energy. So we're not going to do calculations at this point. I will tell you that, so you can relax about that. There won't be Gibbs calculations on this exam. That will be for the next exam. Okay? But you need to understand what Gibbs free energy tells us. Okay? Now, the standard Gibbs free energy for a reaction, if it is negative for that reaction, it indicates the reaction will proceed forwards as written. If the standard Gibbs free energy is greater than 0, the reaction will proceed backwards as written. If the standard Gibbs free energy is equal to 0, the reaction is at equilibrium. And I think I said at the beginning of the term, equilibrium does not mean equal concentrations. Don't forget that. It simply means, equilibrium means that the forward reaction rate and the reverse reaction rate are equal. There's no net change in concentration once you reach equilibrium. Okay? The term in this equation that everybody confuses is delta G 0 prime. Because they want to make it delta G. Because it looks like delta G. If I called it Larry, you probably wouldn't do that. But unfortunately, I don't get to write the rules about the equations. Delta G equals Larry plus RT times the natural log of products over reactants. It would make sense, right? Right? Is Larry the same as delta G? No. Is delta G 0 prime the same as delta G? Well, I don't know. If I asked you right now, are there times when delta G 0 prime equals 0, would you say, how many would say yes? How many would say no? How many yes? How many would say no? How many are dead in their seats? Oh, the dead ones raise their hands. I like this. This is great. So, okay? Yes, of course, there are times when they are equal. Very simple time when they're equal. What happens if the concentration of products equals the concentration of reactants? What happens to the log term? It becomes 0. Then delta G equals delta G 0 prime plus 0, which means delta G equals delta G 0 prime. Or delta G equals Larry, as the case may be. Right? Under standard conditions, products and reactants are equal. That's why we call this the standard Gibbs free energy. The standard Gibbs free energy is the Gibbs free energy under standard conditions. When those two concentrations are equal, those two terms are equal. Okay. Makes sense? All right. I'm going to tell you how an enzyme works using a very simple representation of delta G, and then I'll tell you a joke. You get a song and a joke today. Okay? You can't... I'm not looking for applause. Thank you. All right. How does an enzyme do what it does? Well, an enzyme does what it does not, underline not, by changing delta G. It does not change delta G. It changes the rate with which equilibrium is reached. It speeds the race to equilibrium. Okay? Now I'm going to show you a graph that's going to show what looks like a change in delta G, but it's not. Okay? That's not what I want. It's the next one. Okay? If we look at the free energy of a reaction versus the progress of a reaction, and we look at an uncatalyzed reaction up here, okay, we discover that to go from A going to B, this is the reaction, A goes to B. From A to go to B, there is an energy barrier of A going to B. There is an energy hump that has to be gotten over. That hump can be gotten over in a variety of ways, depending upon the reaction. But one of the humps is the positioning of each other and hitting each other with the right orientation. Okay? In the absence of a catalyst, that hump is shown here. If we look at the delta G, if the free energy before the reaction and the free energy after the reaction, for that A going to B reaction in the absence of an enzyme, we see that the product has less energy. Okay? Therefore, the reaction is favorable. So the delta G for the reaction by itself is a fixed value. In this case, the delta G is negative. Okay? Now, if I do the same reaction in the presence of an enzyme, what I discover is that energy barrier is way lower. The starting point is the same. The ending point is the same. I have not changed the overall delta G using an enzyme. I have simply made it easier to get from here over to here. The analogy I like to give for this is the facts, and oh boy. I better save that for next time. I'll give you the analogy next time. I'll give you the joke now, okay? All right. It's Friday. This is my duck joke, okay? So these three guys go to heaven, okay? And they get up to heaven, and they get to the pearly gates, and they look around, there's these ducks everywhere, okay? Everywhere there's ducks. And the guy at the pearly gates says, whatever you do when you're here, you don't step on a duck. Okay? What the heck, you know? And so they're careful, but after a day or two, one of the guys steps on a duck. And all of a sudden, these sirens go off, and these police come down, and they grab this guy, and they've got this mean old woman. And they put a handcuff on him, and they put a handcuff on her. And they said, for stepping on a duck, you're going to spend the rest of eternity linked to this woman. Man, this is not heaven, right? This isn't good. Okay? The other two guys had their eyes kind of opened by this. You know, they're kind of aware of, well, I'm not going to step on a duck, right? So they're very careful, and they're tiptoeing around, you know, and just being very careful. And they're pretty good for about a month. And one of them steps on a duck. And the same thing happens, and the sirens are even louder, and it gets worse. And the police come down, and they've got a meaner woman. And she's got a frying pan in her hand. Okay? And they handcuff him to her and say, you're going to spend eternity, for stepping on a duck, you're going to spend eternity, you know, handcuffed this woman. And she's beating the guy in the head like this, you know. This third guy, this wasn't what I thought heaven was going to be all about, you know. Literally, he doesn't move. He's afraid he's going to step on a duck because there are ducks everywhere. So he just, you know, very, very careful. He goes for a whole year. He doesn't step on a duck. Okay? And one day the sirens go off. And I didn't do anything, you know. And the police come down and they grab this guy and they got this beautiful, voluptuous woman. And they handcuff him to her and he goes, wow, he said, that wasn't quite what I expected. And the woman says, that's what I get for stepping on a damn duck. Yes, sir.
So yesterday in citation we were calculating this protein charge and all that stuff. So do we need to remember all these structures? Or?
Well, you need. What did I say about those in class?
You said you need to remember any names. Or?
No, I didn't say that. Go back and look at the lectures. You'll see what I said about what you need to know. I didn't say you need to just know the names. You need to know what categories they fit into, and the categories they fit into tell you what's out there. So if they're a carboxyl-containing amino acid, you know they've got a carboxyl group, right? Okay. How you doing?
Good, good.
You like my joke?